One way to get genetic variation in bacteria, and different species can do different versions of these, is called horizontal, or sometimes you'll hear it called lateral gene transfer. There are three main types of that. The first one I want to talk about is the simplest type. This is called transformation. I have a lot of students that don't remember this word. So, when they write all kinds of other trans words. So make sure you know transform, transformation. This should be somewhat familiar to you from Griffith's experiment in 1406. You can take a look at that. It's with the mice that die and don't die depending on the rough bacteria, the smooth bacteria, and then he can transform rough to smooth. In any case, that's in your book in the DNA chapter. So the main thing about transformation here is that it's going to be what we're going to call naked DNA from the environment. And we'll see one way that that could happen. And the whole thing with this genetic variation, how this works is that the DNA that comes into the cell may have different alleles on it, or potentially completely different genes. And so the bacteria that picks up that DNA or has transferred that DNA in future types of gene transfer that we'll look at now has different allele combinations, completely different alleles, and potentially completely different genes, which may give it different kinds of phenotypes. For example, the ability to produce a certain amino acid, so it would no longer need media to grow on with that amino acid. Or resistance to antibiotics, we'll see that later. Or the ability to make a conjugation Pilis, we'll see that later as well. I'm not going to particularly draw the genes that are different in these, but I do have some animations that I will either give you links to or insert here that have a little more detailed version of that. Okay, so I'm going to draw two bacteria to start with. I will draw their double-stranded DNA here. Okay, there's one bacterium, here's another, and they're just in the similar environment potentially. They're not next to each other particularly here. Okay, there's their double-stranded chromosomes, so we should mark that as the bacterial chromosome. And that's true for both of these. In the end, this is going to be a donor bacterium, and this is going to be the recipient. There's no real reason that this is the donor, but the recipient does have to be what we're going to call competent, so it has to have certain proteins on its cell membrane through its cell wall, and we can call those DNA binding proteins, so it's just going to have those. Right there, and that's what we call competent, meaning that it's going to be able to take up the DNA from that's released by the other bacteria. So there's our starting scenario. All right, so now what happens basically is that this bacterium, let's say, dies. And what happens is the cell wall and cell membrane are going to break down, and the DNA is going to be degraded. And so you'll have little fragments of DNA around there. So here's a chromosome fragment. Okay. And then you'll have those fragments near, they'll be in the environment and end up near the other bacterium that has this DNA binding protein. And if it contacts that fragment of DNA in the environment, it can actually take it up. One of the strands will be degraded by parts of that parts of the system in there, enzymes, and then the other strand will end up inside of that cell, and this cell still has its chromosome as well. Now here's the thing, is that these two chromosomes have to have some sort of homologous sequences, meaning that they are very similar. And that's why we're going to have pairing between the initial chromosome, and you'll have some pairing with this fragment because of those similar sequences. And then machinery in the cell may cut and basically kind of flip in this homologous sequence, and then you get that original sequence of the chromosome that gets flipped out. And this is through machinery. This should remind you a little bit of crossing over in eukaryotes and meiosis, but it's not the same thing. But we do call this homologous recombination. And then the excised DNA is degraded. And there are one of two things that happen. So because there are different sequences on there, the bacteria cell will detect that and will repair those differences. So one possibility is that the interior strand is used to repair the DNA, and so what you'll end up getting actually is the same sequence that you started with. So this would be a non-recombinant bacteria in the end. It'll be exactly the same. So that's one possibility. Or another possibility is that the exterior strand with that different DNA sequence here is used to repair. I don't want to make it look double-stranded. There we go. Is used to repair, and so you'll get a cell with that new DNA sequence, and it may carry a different gene, and so we would call this a recombinant bacterium. And that's the one that would now have different alleles in different combinations and or potentially completely different genes. And so obviously now the bacteria is genetically different than what it started with, which is what we call genetic variation. Another possibility for horizontal or lateral gene transfer is called transduction. Okay, transduction. And this is involving viruses. So actually it's accidental, and I'll say viral transfer of bacterial DNA. Now remember that the type of virus that infects bacteria specifically is called a bacteriophage or bacteriophage, or just for short, phage. People do call it phage. So that's what's going to be in action here. It's not just any bacteria or any virus that does this. All right, so we're going to start with one cell here that we're looking at. This is ultimately the donor cell, but there's no particular reason it's the donor cell. It's just because it's the one that gets infected with the virus. So there are viral particles that are in the environment, and they carry viral genomes. And in this case, I'm going to draw, I'll draw double-stranded DNA still. Okay, so there's our bacterial chromosome. Okay, and this bacterium has these proteins, complexes on its cell membrane, cell wall that allow the virus to basically poke through the cell wall and inject its DNA, or I should say genetic material, into the bacterial cell. This is just normal viral infection, nothing different here. And then, as is typical, now the viral DNA will be inside. That will replicate using the cellular, the host machinery, and the host molecules, and the DNA will fragment. A lot of it will be degraded. Those nucleotides will be used to build the viral DNA. And then we have the empty phage here. Okay. So now we have an infected cell, and that cell will produce viral capsid proteins, which will then self-assemble in the cell. They don't have any signals per se as to what to take into their cell. So most of the genome in the environment is going to be viral, but occasionally, by accident, one of the viruses picks up some chromosomal DNA from that original bacterium. So we now call this a transducing phage, because it has bacterial DNA by accident. Okay. So just as is normal with an infection, so this would be lytic type phages or temperate phages that are going to rake open the cell. I'll draw this here. So it lyses, then we release the viral particles. Most of those have viral genome in it, which can then go infect other cells. That would be these two. But some of them, or a few of them, there we go, have the bacterial DNA. And they don't, the virus, quote, doesn't know the difference. They don't have really much sensory features, so it'll just go and do its normal thing and infect another cell, bacteria. And this one would have to have the proteins on the surface for that. It's not for the virus to bind to, but it can. And then basically it'll insert that DNA into the cell, and this is going to be a different cell here. So I'll draw a different genome. So this is another bacterial cell, now infected with that virus. So just like a virus normally would do. But it's not viral DNA, so it's not going to trigger the breakup of that chromosome, the degradation of the chromosome. And if it's homologous to that chromosome, similarity in sequence, it'll basically pair with that. And there's machinery in the cell which can recombine the DNA. And so now you have different DNA flipped into the genome. And so that other DNA, the double-stranded host DNA or recipient DNA, will be degraded. And so you'll end up with a cell that has new DNA in it. It doesn't know the difference. It's bacterial DNA. And that could result in different phenotypes, and therefore there has arisen some genetic variation. The last type of horizontal gene transfer that I'm going to talk about is called conjugation. Sometimes people call this bacterial sex, but I don't really like that terminology. I don't find it to be very accurate, so we will just call it conjugation. And you'll notice you have the co here, which means together. So this actually is the direct transfer of bacterial DNA frequently a plasmid. And I should mention that plasmids can be transferred via transduction as well with the viruses, but a lot of times we see it with conjugation. Direct transfer, and in order to do this transfer, the cell has to be what we call F plus, which means it contains the fertility factor, which will be on a plasmid, which will be the F plasmid. We can also see that as a conjugative plasmid. Okay, so if I draw the F plus cell, I'm going to draw it big first. I'll draw it with the Pilis, and I'll draw the Pilis more detail in a moment so you don't get the wrong idea about it. Here's the bacterial chromosome. Contains all the genes to live as a bacteria. And then we have the F plasmid, which is smaller. Those plasmids are pieces of DNA in the cytoplasm that get replicated and passed on to the next generation. Like I said, conjugative plasmid, we also might hear that. And then this is the conjugation, or you might hear it called the sex, Pilis. And just so you don't get the wrong idea about what a Pilis is, it's not even an extension of the cell membrane like a flagellum in a eukaryote would be. It's proteins embedded in the cell membrane, and then a tunnel of proteins, basically, that's extension from that. So we have the cell membrane and cell wall, and then part of it is embedded in it are this protein complex here. It's a bunch of different proteins. And then from that, we have a protein structure that extends from there, and it's basically a tube. It has a bunch of alpha helices. I have a crystal structure of it that I'll embed or link to somewhere. And it just sticks out there. So it's not like an extension of the cell like it looks like in drawings. Sometimes it's proteins embedded in the membrane and then extending from that. And this part with the Pilis part that is the part that retracts and builds, so it can get longer and shorter when it contacts the other bacterium. And just so you don't get confused, what we're looking at, this is the bacterial chromosome. And then this is the plasmid, like I drew in the previous schematic. Okay, so let's start with our two cells again. I'll just draw this with the Pilis already formed. Use blue. And I'm going to use two colors here because you need to differentiate. This is all the plasmid though. And here's the other cell and its chromosome, double-stranded DNA. So this one is the F plus with the F plasmid and the conjugation Pilis, and this is the F minus. You see that it does not have a plasmid. This is just its chromosome. And so this Pilis contacts this other bacteria, basically punctures the cell wall and cell membrane, gets through there. I'm not going to draw the chromosomes at the moment. And just remember, this is the conjugation Pilis or sex Pilis. Then it will retract and bring that bacteria cell close to it and form a little tunnel through there. So this part here, the actual connection between the two cells, is called the conjugation bridge. And it's actually an opening between the two. So now we have this bacterial chromosome with this plasmid, the F plasmid. We have this bacterial chromosome over here. Now this can happen between the same species or different species. It just depends on the scenario there. Okay. Then we get the actual transfer of the information. So there are special genes that are on the plasmid that allow this to occur. You'll learn about those in future classes. I'm not concerned about it. What you need to know is that there is an enzyme in here that can nick the DNA. Basically, that just means to break one strand of the backbone through hydrolysis. And then there's proteins that can transfer that DNA from this donor cell to the recipient cell, the F- cell. So it still has its chromosome, and this one still has its chromosome. And it's just the plasmid copy. It's being transferred. Now notice, not the entire plasmid. Sometimes people get confused and they say transfers the plasmid, but that's not actually the case. It's transferring one strand of the plasmid at this point. Then that one strand will now be in the other cell. So there's one of the original strands. Here's the other strand, and it will circularize and reseal that nick, which means that backbone is now one normal continuous piece of DNA. So I've got the double-stranded chromosome, just like it was, and now there are enzymes that can synthesize the other strand. Okay, and so now you have a plasmid, it's an F plasmid, and now you have another one in this cell. So now really you have the original F plus cell, and now you have a second F plus cell. So then those two cells will release that conjugation bridge, and now there'll be two separate cells. Each of those actually has the ability now to make its own Pilis. And just by those proteins, because now they both have the F plasmid, or a conjugative plasmid, but we call it the F plasmid. Okay, because now we have two F plus cells. So that's one way conjugation can occur. Another conjugation scenario, and like I said, there are multiple ones, but this one I'm going to draw. Okay, so here's our F plus cell. It has our DNA in it, and it's going to have, I'll draw it like this time. This is our conjugative or F plasmid. And then this time it also has this other plasmid here, which is going to be a mobilizable plasmid. This can be multiple things, but common when we talk about is the R plasmid, which R stands for resistance, because this transfers or contains genes that allow the bacteria to survive antibiotic treatment. Remember, it's not, quote, for that, but it gives the bacteria some kind of mechanism. So, for example, transfers a gene that codes for a protein that can pump the antibiotic out of the cells, the bacteria. Or it codes for a protein that prevents that prevents the antibiotic from attacking the ribosomes and destroying those, if that's how it works. So here's our donor bacterium, and there's our recipient bacterium. And just remember, this is our conjugation pilus here, formed of proteins. So now we have a similar sort of scenario. I'm not going to draw the DNA yet. We have the plasmid, and it basically pokes through that cell wall and then pulls those bacteria cells together, forming this conjugation or mating bridge. Same thing as before. And then this time, part of this mobilizable plasmid is going to get nicked, and then one strand will get transferred into the other bacterium. Both of them still have their genome. And this one also has the F plasmid still. Same thing's going to happen. I can think it kind of happens simultaneously, but we're going to draw it like this. Genome, F plasmid, mobilizable plasmid, synthesizes its interior, or the second strand, I should say. So now we have two complete ones of those. Then we end up the cells breaking that mating bridge, and we have two cells, each of them with a mobilizable plasmid. Let's say the R plasmid. So now we have two cells that are resistant to the antibiotic. One of them is F plus, so it can keep passing that on. The way I've drawn it here is the other one is not still. So we have an F plus resistant cell. We have an F minus, but now resistant cell. And so this one can go on and form more pili. I'm sorry, that's a distorted cell, but it can spread that resistance factor and more. This one, the way it is now, can't spread this resistance factor. However, if a bacterium with a fertility factor were to conjugate with it and pass on this fertility plasmid, the F plasmid, then you get that cell turning into an F plus cell with the resistance plasmid. Then it could make the conjugation or sex pilis and be able to transfer that antibiotic resistance. And that definitely happens. And remember, when the cell replicates by binary fission, it's going to pass on all its plasmids as well. So all the progeny of both of these cells, when they divide, will have the resistance plasmid and the resistance gene in there. So they will all be resistant to the antibiotic in this case. There are also conjugation scenarios where you have the F plasmid integrated into the genome instead of its own plasmid structure. So the F factor is in the genome of the bacteria. Now we call those HF R cells, high frequency of recombination. I'm going to put up an animation showing you that instead of drawing it here, but you can see that it's transferring the F plasmid plus a little bit of the bacterial chromosome, so that's another way to generate variation in your bacteria. So overall, these are the three main ways that horizontal or lateral gene transfer can generate variation in bacteria because they do not sexually reproduce and the mutation rate is low. And so this is one of the main ways that bacterial populations gain diversity and so therefore are better able to survive insults in the environment, not because they change to the environment, but because there's genetic variety in the population to begin with. This is also one of the ways that antibiotic resistance spreads very quickly in bacterial populations, which obviously can be a really big deal for the treatment of bacterial infections in humans.